For thousands of patients, survival is a matter of timing. They wait for a donor organ that often will not arrive before their body fails. This chart shows the reality of the global organ shortage. There is a widening gap between the number of available human donors and the millions of people who need them to live. Because human donors cannot meet this demand, researchers have spent decades looking for a way to source organs from outside our own species. The most viable candidate is the pig. It wasn't selected for convenience, but for anatomical compatibility. Its internal organs are remarkably similar to our own. To bridge the gap, medicine is attempting to turn animal biology into a scalable, reliable supply chain for human patients. A pig's heart, liver, and kidneys are close enough to a human's to theoretically sustain our circulatory and filtration systems. However, the human immune system functions as a biological fortress. It is hardwired to identify and ruthlessly attack any tissue it doesn't recognize as self. This diagram shows a cross-species transplant reaction. The body identifies the animal tissue as an infection, triggering a rejection that destroys the organ in minutes. It starts at the surface. Molecular markers act as alarms, signaling the immune system to assault. Traditional surgery cannot stop this reaction. To make the body accept the transplant, scientists had to find a way to rewrite the biological code of the animal tissue. Since doctors cannot safely disable a patient's immune system, they turned their attention to the pig, using genetic engineering to modify the donor rather than the recipient. This visualization shows the pig's DNA sequence. Researchers mapped this code to find the exact genes responsible for producing those molecular alarm signals. Using gene editing tools, they silenced these markers, removing the biological flags that trigger an immune response. This creates an organ that is effectively cloaked. It functions as a pig organ, but no longer broadcasts signals leading to rejection. This turns the animal into a specialized living vessel, engineered specifically to survive within a human host. By modifying the genetic source code, researchers built a biological workaround that allows a foreign organ to bypass our most aggressive natural defenses. The first human trial involved a patient with no remaining medical options. Facing certain mortality, they agreed to test whether this engineered biology could actually function in a person. The engineered organ was transported into the operating room, a physical manifestation of years of laboratory research. Under the theater lights, the surgical team began the precision work of connecting the animal tissue to the patient's circulatory system. They carefully sutured the vessels, establishing the lifelines that would determine if the human body would accept or resist the new organ. In the moments after the connection was made, the theater went quiet. Everyone waited to see if the modified organ would hold against the patient's immune system. Then, the organ flushed with color. It didn't fail. It began to beat, to filter, and to stabilize the patient without triggering an immediate attack. In a matter of hours, years of genomic research were validated as a functioning biological system inside a human host. This success has massive implications for global healthcare, moving the discussion from experimental theory to practical application. This flowchart illustrates the shift. We are moving away from a system that relies on rare human accidents for supply toward a scalable model where organs can be produced through engineering. The goal is a healthcare landscape where hospital waiting lists are eliminated and no patient dies because a donor wasn't available. By addressing the root cause of the shortage, scientists are attempting to change the math of transplantation from a resource crisis into a manageable process. But the ability to merge species creates deep societal tension, moving the focus from medical success to ethical inquiry. Religious leaders and ethicists are now questioning the boundaries of human life and the long-term consequences of relying on engineered animal parts. It forces us to redefine what it means to be human when a part of our survival depends on the DNA of another species. While the debate continues, clinical trials are expanding. Each patient who survives provides critical new data on how these engineered systems perform over time. The initial trial was successful, but as we move toward a future where organ scarcity might be solved through engineering, we are only beginning to calculate the ethical price of that survival.